In this video, we will discuss about global analysis of glycoprotein chaperones, uh, or more specifically intracellular chaperones, that is Calnexin, Calreticulin, and MOGS, that reveals the this lectin's role, intracellular lectins roles in regulating ER stress and unfolded protein response. So, what is the motivation? We can say idea or motivation behind this project is recently published study the recently published study in Hematol on Call that shows the that shows about this myeloproliferative cancer. In myeloproliferative cancer, different kind of mutations are predominant, and one of the predominant mutation in this different kind of myeloproliferative cancers is JUG2 mutation, and the next predominant mutation is this calreticulin knockout that we can find in ET and PMF blood type cancers. In this ET blood type cancers, the disease occurs because of high platelet formation, and in the case of PMF, the disease caused because of low blood formations. And the role of calreticulum in this kind of disease is that this calreticulum might migrate from its endoplasmic reticulum to this extracellular matrix and interact with this MPL receptor, and this disrupts the function of this MPL receptor. That is, this MPL receptor helps in disrupt the function of MPN receptors, such as that is MPN receptor helps in this thrombobiotic signaling within the cells, thrombobiotic production signaling within the cells. And so let us start with the background. Since this calreticulum, we will study, our focus is on the calreticulum protein. So we will study the background where calreticulum plays an important role, that is calnexin and calreticulum cycle in endoplasmic reticulum. So what happens in this case is that the N-glycan precursor will enters the endoplasmic reticulum, and this addition of this three monosaccharide addition of these three monoglucosylated saccharide takes place. And after that, during the co-translation of the proteins, this N-glycan precursor will add to this newly translated proteins, and after that the MOGS, it will interact with the MOGS and GANAP, so that this monoglucosylated one of the monoglucosylated monosaccharide will be trimmed off by MOGS and this GANAP, and then the newly translated proteins will then interact with this calnexin and calreticulum, so that they can acquire their native structure. And if the proteins acquire their native structure, they will move to the Golgi for further modification and glycosylation, and otherwise they will move to the cell degradation. So our area of study is that we have knocked down this MOX knockout and calnexin knockout and calreticulum knockout, and then study the global analysis or global changes within the cells, so that we can predict how important are each of these intracellular chaperone or glycosylation processing proteins within the cells, within the cell functioning. So what is the plan that we have used? The first one is this: we took the hex cells and then prepare the CRISPR knockout. Then we study this proteomics study, and this lectin binding study, and whole glycomics profiling, and finally we end this study with the cell cycle and apoptosis. So in the case of proteomics study, so first, in the case of proteomics, we have analyzed the label free we did the label-free analysis of differential expression of the protein, and then analyze the upregulated and downregulated proteins, and try to find out what is the common things between these upregulated and downregulated proteins in different knockout. And in the case of upregulated, we have find out that proteins that are related to UPR or unfolded protein response are more predominantly upregulated compared to the other proteins. And apart from that, the cross-contamination proteins are also upregulated, that is ATP13A in Calnexin and Calreticulin knockout. So this indicates that there could be a cross-contamination of the proteins from endoplasmic reticulum to mitochondria in the case of Calnexin and Calreticulin. So they might have a role in preventing those kind of contamination. And when we analyze these downregulated proteins, we find out that some of the glycosylated related protein, both the N-glycosylated proteins and the O-glycosylated proteins are downregulated. So this indicates that this Calnexin and Calreticulin this substantiates the point that Calnexin and Calreticulin plays a role in N-linked glycosylation and also the protein folding. And when the proteins are not able to acquire their native structure, they will divert it to this ERAT pathway, that is endoplasmic reticulum associated degradation. Thereafter, since from the proteomics study, we come to a point that there could be an unfolded protein response that could have triggered in the case of this absence of glycosylation processing protein. So we decided to check the unfolded protein response. So this is just a brief overview of the unfolded protein response. So what happens in the case of UPR is that three signaling pathways get activated, that is ATF6, PERK and IRE1. And when we check the literature and we are able to find out that there is a fluorescence reporter available for this ATF4. So we decided to move forward to check whether there is a UPR response, exist in different knockouts of the cells or not. And when we check that with the help of this ATF4 fluorescence reporter, we can see that there is a UPR response in all the UPR response in the knockout, but this UPR response is not at the same level. All the knockouts have different degree of UPR response that can be observed from the differential fluorescence signals from all the knockout, different fluorescence signals from all the knockout. When we quantified this image, we can see that the Calreticulin has the highest ER stress compared to the other knockout. And in order to validate these results, we check these fluorescence signals through other instrument, that is flow cytometer. In the flow cytometers also we can see that the fluorescence from the Calreticulin is very high compared to the other knockout, and the Calnexin and MOGS ER stress are almost at the same level, whereas we do not see any ER stress signal in the dual knockout. Thereafter, we also check one of the signature protein that is produced during the ER stress, that is the CHOP protein, and we can see the results are in line with our fluorescence observations. That is, the Calreticulin knockout has the highest CHOP production, and the MOGS and Calnexin knockout is CHOP production is almost we can say comparable. And for the dual knockout, we are not able to see at this point, but for the dual knockout also we can the signals are almost negligible, and that is in line with our fluorescence assay. And thereafter we decided to check this we decided to move forward and check the change in the glycomix, as this Calnexin and calreticulum cycle are uh, known for the initiation of end-linked glycosylation, also known for the initiation of the end-linked glycosylation. And first we check this cell surface glycomics study, and in this study we did this study on the 23 different lectins, and we can find that out of these 23 lectins, only 4 lectins show significant upregulation or when compared to the wild type, and all these 4 upregulations are found out in the case of calreticulum knockout. So this could hint towards a point that in the case of calreticulum knockout, there could be a more deviation in the protein native structure, and that might lead to different glycosylation profiles. And when we did the whole cell glycomic study, and we can see the complete or more big picture of the that is more big clear picture of the intercellular functioning of the cells. For example, in the case of this dual knockout, we can see around 38 unique glycan structures. So this could be possible only when there is a quality control loss occurs in the dual knockout, and most of these unfolded proteins might move into the cytosols, and instead of diverting them towards the ERAT pathway. And afterwards we can see that in the case of MOGS and Calnexin knockout, in the case of MOGS and Calnexin knockout, number of unique glycans are almost similar, around 10 and 11. So this indicates that quality control in the case of MOGS and Calnexin knockout is almost comparable. Thereafter, the least number of unique glycans we have found for the calreticulum knockout. So from this whole glycomic study and also from this cell surface study, we can make a point that least number of unique glycans are found in the case of calreticulum knockout. So this could mean that quality control in the case of calreticulum knockout is quite high, and the unfolded proteins that are diverted are mostly diverted to the ERAT pathway. And one more signals that we have observed for this point is that the proteins that are involved in ERAT pathway are upregulated in the case of calreticulum knockout. And finally, we check this apoptosis and cell cycle. In the case of this apoptosis assay, we can see that almost all cells behave similarly, and there is no significant changes in the apoptosis of the cells compared to the wild type. When we did the cell cycle assay, we find that there are some changes that are occurred. There are some significant changes that are found in all knockouts, and in all knockouts in terms of this G1 phase, whereas for the S phase we can find that in the case of calreticulum MOGS knockout there is a upregulation in the case of S phase, and this could indicates that calreticulum and MOGS knockout might interact with the proteins that are essential for this that plays a role in this G1 phase and S phase cycle. And what are the main conclusions that we can get from this study is that Calnexin and calreticulum cycle plays a role in maintaining the homeostasis of the glycoprotein within the endoplasmic reticulum. So this, in other terms, if this Calnexin and calreticulum function normally, then we will not see an overcrowded endoplasmic reticulum that might trigger ER stress. And other things that we have find out is that since the calreticulum has the high ER stress, one of the reasons could be that the calreticulum has a broad specificity. And another thing that justify this uh, observation is that this calreticulum is a ER lumen proteins, so its availability to the different proteins is very high compared to the calnexin, as the calnexin is a ER lumen proteins. So, and other things that we have find out is that quality control hierarchy in the endoplasmic reticulum. So in this case, what we have find out is that calreticulum knockout has the highest quality control. Thereafter, the MOGS and Calnexin knockout quality control is almost comparable, and then the dual knockout quality control. So this could be this is the structure of the calreticulum arm and lectin domain that plays a important role or interaction of these newly folded proteins, and at the same time this domain also interacts with these other proteins from other families such as PDI family, and that helps that form a complex along with this calreticulum knockout, which helps the proteins to acquire their native disulfide bond. So that's all for this video. Thank you for your time.